Now you know about all the major animal phyla but one, Chordata, which is the phylum that humans are in. One of the big differences between the groups we have covered so far and the ones that we will spend this entire unit on is the presence of a backbone or spine. This is a phylogeny for chordates. I want you to notice that this is a very diverse group that includes fishes, frogs, snakes, birds, and mammals. That diversity deserves more than just one lecture to cover. You can see that most are considered vertebrates, but two are not, and these are the taxa that I want to cover in this short lecture. These are animals that evolved before vertebrates and showed us how life transitioned from animals like echinoderms to ourselves. This lecture will cover sea squirts and lancelets, neither of which you have likely heard of before. The next lecture will cover hagfish and lampreys, often referred to as jawless fishes, cartilaginous fishes, which include sharks and stingrays, and bony fishes, which includes most fish. We will then go over amphibians, reptiles, birds, and mammals in separate lectures. So to start, how do we define the phylum Chordata? What do all these animals that look very different have in common? First of all chordates have gills when they are embryos. That's right, you had gill slits when you were in your mother's womb. No, this does not mean a mutation would allow a baby to be born that could breathe underwater, because these gills become your ears. But sometimes the ears do migrate incorrectly and babies are born with their ears on their throat. All chordates have a dorsal hollow nerve cord, which is a tube that runs along the back as the central stem of the nervous system. In the group that I referred to as vertebrates, this develops into the spinal cord, which is not hollow. Lastly, like echinoderms, our anus develops first, which means that we are in the groups referred to as deuterostomes. There is a second tube that runs down the back called the notochord that provides support, and this is what in vertebrates becomes the spine or backbone. One tube basically wraps around the other, such that the vertebrae protect the inner spinal cord. Lastly, all chordates have a postanal tail during development, including humans. And yes, sometimes a human baby is born with a fully developed tail, but for most of us, the tail is reabsorbed. Believe it or not, these animals have all those features.
Tunicates, fixed to the rocks, still laboriously siphon the waters for food. They may lack backbones, but they are related to us.
An often overlooked sea creature, the sea squirts or tunicates are a fairly common group of 3,000 species. How could they be related to fish? They look more like sponges or jellyfish. The similarity becomes clear when you look at their development. Their larvae look like tadpoles with a head and tail, but more importantly, have all the characteristics of chordates. This larva swims around until it finds a spot to attach, and it metamorphizes into an adult tunicate, which is a sessile filter feeder. It has an incurrent and excurrent siphon similar to sponges and bivalves. Another group looks a bit more fish-like. Lancelets, also known as Amphioxus, are marine animals that also filter feed. They burrow into the sand and use tentacles around their mouth like sea anemones.
This diminutive creature, Amphioxus, tells us volumes about our own evolution. Without eyes, ears, or jaws, this is a very simple organism. Yet it has some surprising things in common with us. Amphioxus have some body equipment oddly like our own. Amphioxus has a nerve cord which relays commands from the brain. It has gill slits, just as human embryos do. It has segmented muscles that allow it to move, but the real innovation was the notochord, a stiffening rod reinforcing its shape. This was the precursor of a backbone. We carry vestiges of a notochord, the discs in our spine. Our spines place us in a special group of chordates called vertebrates. The backbone is the crux of every vertebrate body. It is the central scaffolding of the skeleton. All vertebrates have common ground with Amphioxus. But how did we become so much bigger, so much more complex? The same genes direct development in Amphioxus and every other animal with a backbone.
Studying the DNA of ancient organisms is a window into our distant past. And if you look hard enough, you can find some creatures today that haven't changed much in hundreds of millions of years. This coast is home to a relative that shows us how ancient the genetic recipe is for building our brain.
Yeah, how do you know where to look? Well, certainly want to go deeper into this.
Meet Peter Holland, head of zoology at Oxford University in England.
Right, okay. Well, we could start trying around here.
X marks the spot?
X marks the spot. Let's give it a go.
Okay. Okay, so what do I do here?
Just shake it. You got one. There's an Amphioxus.
We got any? Whoa, look at that little guy go. Hey!
Just dip it in the water again, you'll see it, it'll flick around.
Well, they really flick.
There you see. There he is.
Hey, that's beautiful. Look at that. Which is the front?
It doesn't look like much, but this tiny creature, called Amphioxus, has much to tell us about our own brains.
That is cool. Okay, that ranks as cool.
Okay, so let's take a look at a couple of these.
So I brought this from the anatomy lab in Chicago. This is a human brain. And when I look at this, I don't find any obvious similarities to that.
Deep in the genes of this animal and the development of this animal, and deep in the genes of us and the development of us, there are the similarities. So if you just take a look at that, he's just moved.
Oh, he
They're
twitched
pretty
away.
active.
Yeah, he's
They're
wow.
pretty active.
Holy cow. Oh, that is beautiful. God, they're so clear too. You can see right
Yeah,
through them.
yeah,
It's amazing.
yeah.
Amphioxus lives in the sand of the ocean floor, filtering algae out of the water. They have a simple nerve cord that runs the length of the body, which ends with a tiny swelling invisible to the naked eye.
If you look at the front end of this animal, you don't see it all expanded into a large skull or brain region. It's just pointed.
We might not have similar brains, but what we do have are similar genes. To make the forebrain, midbrain, and hindbrain, you need a series of control genes to tell the cells within the developing brain where they are. They map out the brain's 3 basic regions. Versions of the same master genes that help form our own brains are also active in Amphioxus, but here they're simply building the primitive nerve cord and the tiny swelling at its front. This means that the genetic roots of our own brains can be seen in creatures that first arose over 500 million years ago.
I think it gives us a glimpse into where our brains came from, into the basic organization of the brain of our ancestors.
I mean, I find that mind-blowing.
He's jumped off,
he's hot.
guys. Sorry.
He's going
We have an
Tiktaalik
escapee,
on us.
sorry.
He's on land living. He's gone rogue.
Yep, he's gone for the next transition.
Exactly. He's in the Devonian now.
The 30 or so species of lancelets have several more features in common with us than tunicates. They have muscles that run alongside their body, called myomeres, a primitive liver for secreting digestive enzymes, primitive eyes, a cartilaginous rod for support, and a head with a brain. They may not look like much, but they are one of your closest living ancestors.